My name is Samhita Pokkunuri, and I'm currently a ninth grader attending Old Bridge High School in New Jersey. My research project is developing a novel graph neural network framework for drug discovery. I also use this framework to identify new drug targets for treating glioblastoma. The discovery of new drugs can take many years of research and costs billions of dollars. A significant amount of time is spent to find the perfect drug molecule that can perfectly bind to the disease, causing a target protein molecule. With the amount of data available, biological computational methods can effectively help reduce the timeline of initial drug discovery by predicting drug-target interactions before starting the experimental methods. Over the past few years, many deep learning methods have been proposed to learn from protein sequences and molecule structures using protein-protein and drug-drug associations. However, existing computational methods have severe limitations, resulting in low prediction performances of drug-target interactions. To combat the issues with current methods, I formulated my research question. Can I create or model a deep learning framework to better predict drug-target interactions and thereby accelerate the initial phase of drug discovery? In this research study, I built a machine learning model that can take protein and drug sequences as inputs and give a confidence score of their interactions as outputs. Before feeding protein sequences and drug molecules to the model, they need to be encoded as numeric vectors. For proteins, k-mers are a classical and effective method for protein embeddings. K-mer features can describe the type and number of amino acid functional groups, which are very important in the prediction of drug-target interactions. For drug molecules, fingerprint is a very efficient technique in drug discovery and virtual screening. In molecular fingerprints, the topological information of molecular structures are encoded as a vector, which can well represent a drug molecule. Well, after extracting the protein and drug sequence features, how can we predict the interactions between them, especially those unconnected protein-drug pair candidates? To solve this issue, I used hypernodes as neural connections, which act like neurons in our brain during memory replay. Hypernodes are virtual bridges between proteins and drugs that capture the relationships between them. It makes my model learn high-quality drug-drug and protein-protein association information from the given data and further improve the predictive performance of drug-target interactions. A graph neural network is constructed for each drug-target pair and the set of hypernodes as nodes of the graph. The edges are then calculated using the cosine similarity. The output of the graph neural networks is the embeddings of protein nodes and drug nodes are fed to a connected layer. Finally, the sigmoid activation function is applied to classify whether the protein-drug pairs have interactions. I compared the performance of my model with other deep learning methods using the same binding DB dataset, a publicly available database for candidates, drug targets with ligands that are small drug-like molecules. I did not see a significant difference between my model and others for the area under the ROC curve. However, the accuracy of prediction values was much better with my model than other models by a large margin. I would attribute this to the hypernodes introduction that let the model learn the interactions between drugs and targets using deeper interaction rules and predict drug-target interactions better. I also observed that the predictive performance of the model peaked at 32 hypernodes, which made me accept that at some point, too many hypernodes would overfit the model. So to test the real-world validity of this model, I chose to run it to predict drug-target interactions on glioblastoma. I used a bioinformatics and network analysis to identify the common differentially expressed genes across 6 glioblastoma datasets and was able to identify 8 highly connected hub genes that might be implicated. I then used these 8 protein sequences as one input, along with the binding DB dataset as another input to my model. The model was able to identify 17 drug targets for glioblastoma. 2 of these, prazosin and temozolomide, were identified by my model with a confidence score of 0.98 and 0.96. Therefore, this case study on glioblastoma suggests that the model can be used to accurately predict protein-drug interactions. Although my case study is focused on glioblastoma, this comprehensive machine learning model can also be used to predict drug-target interactions for any disease, if we can identify the proteins implicated in that disease. Thank you very much for listening.